as the team lead for the science component of our project, my name is Rebecca Sutton, as Melissa just said, and I'm introducing key members of the very broad science team that undertook this major study we're presenting to you today. All right, I'll get up here. I'll repeat that. So my name is Rebecca Sutton. As Melissa said, I led the science work, and I've got members of the team here before you. This was a big, broad team, so these are key members who are going to be able to provide the high-level findings that we're hoping will inform the discussions on solutions this afternoon. If you want to dig into the report, it is now up on the website, 400 pages. So we're boiling it down to the high-level findings that are really going to be helpful as we move forward. So you're going to hear from Meg Sedlak on objectives. She's with SFEI. We got Carolyn Box from Five Gyres on the surface water data. Diana Lynn's going to be talking about our sediment and prey fish. And Alicia's going to be talking about stormwater and wastewater, Alicia Gilbreth. We've also got Alice Zhu, who will be representing the analytical methods from Chelsea Rockman's laboratory at University of Toronto, so she can address any very technical questions that come up after we review the high-level findings. So I'd like to introduce first Meg Sedlak to introduce our study.
Thanks, Becky. Is this okay, or should I use the mic? It's good? Okay. So I'd like to acknowledge my colleagues who, we had a whole team of people at SFEI working on this project. We also were working with Five Gyres and then University of Toronto. I'm going to talk about our 6 study objectives, a little bit about the vocabulary that we use on this project, and then a little bit about the study design that we developed to answer our 6 objectives. So one of our first objectives was to improve field and laboratory methods. We developed robust methods for collection of microplastics in the field. And at the same time, Dr. Chelsea Rockman and Alice and her colleagues at University of Toronto developed seminal and novel methods for analyzing particles and microplastics in the laboratory. The second thing that we wanted to do was to identify baseline conditions in surface water, prey fish, so small fish, sediment, to understand the magnitude of the issue and to be able to track future trends. Thirdly, we wanted to understand how microplastics were getting into the bay, so we looked at 2 pathways. We looked at treated wastewater effluent, and we looked at stormwater. Thirdly, we wanted to develop a transport model that would stimulate the transport of microplastics through the bay and out into the coastal ocean. This work was undertaken by our colleague, Dr. Rusty Holloman at University of California, Davis. Rusty called me yesterday, and he said, I'm really terribly sorry, but I cannot make the symposium tomorrow. I have an urgent deliverable. And so instead, we have Don talking. But Rusty's deliverable was born yesterday, little Arthur. So he's at home working on that, and we'll have Don talk about the transport model. So we used all this great science that we learned to inform policy options and solutions, and my colleagues at Five Gyres will be discussing that this afternoon. And then lastly, we wanted to disseminate this information really broadly within the scientific literature, so manuscripts, technical reports, communication things like a fact sheet, and we have a film coming out as well. So now I'm going to jump a little bit and talk about vocabulary, because I think that's really important in this field. So what are microparticles? You heard several people say today they're things that are less than 5 millimeters or less than the size of a popcorn kernel. Here's a photo of a water sample from San Leandro Bay, and you can see up in that top corner a couple examples of macroparticles, red plastic drinking straw, some foam bits. If you squint a little bit, you can see some examples of microparticles, like these smaller particles. So we would ship these samples up to the University of Toronto. Their analysts would clean up the samples, and then they would start to, using a dissection microscope, pick the little particles out and plate them onto a Petri dish. And from there, they would use visual observations to classify them in terms of the shape and to count them. And until very recently, this was the state of the art within the field of microplastics. But one of the challenges is, as you can see in the lower right-hand corner there, is you get a blue fiber. And visually, you can't tell whether that's a blue cotton fiber or blue polyester fiber. So one of the things that we strove to do on this project was to use sophisticated methods to analyze the particles, getting at this question of not all microparticles are microplastics. So up here we have an example of a pellet. Alice would run this on the instrument. She would get a spectra or fingerprint that allows us to understand the chemical composition, and she would say it's polyethylene, which was fantastic. One of the challenges is with fibers and some of the other particles that we looked at is that they contain dyes, and that dye can make that fingerprint or that signal be somewhat blurred, and we can't tell the underlying composition of that particular particle. And so for those particles, we refer to them as anthropogenic unknown, meaning that we know that they're man-made, we just can't determine the polymer type underneath it. So what do microplastics look like? We see fragments from larger objects like plastic water bottles. We see fibers from the clothing we wear, as well as materials that are used in industries. You heard this morning of Mark and a couple other speakers talking about nurdles that are pellets that are used in industry. We also see microbeads that are used in personal care products. We see films from single-use bags, and we see foam. And it's a myriad of plastic types, so polyethylene, polystyrene, polyvinyl chloride. It also includes rubber, which is a polymer. So the morphology and the plastic type helps us to understand some clues as to possible sources. So when we see a polystyrene fragment in the surface water samples, we can start to infer possible sources of single-use polystyrene foam packaging containers. So now I'm going to quick flip through to our study design that we developed to answer those 6 objectives that I mentioned earlier. Carolyn's going to talk about the work that we did monitoring surface water in the bay and the open ocean. My colleague Diana will talk about these teeny tiny little preyfish and sediment. And then my colleague Alicia will end the session talking about treated effluent and stormwater, 2 of the pathways that we looked at. So with that, Carolyn, I will turn over the rein to you.
All right. Hi, everyone. I'm Carolyn. I'm at 5 Gyres, and I'm really excited to be standing here today to present the surface water results. I led up the surface water sampling, and it's pretty exciting. So our field work, like Meg said, covered the entire San Francisco Bay, all the way from the north in Sassoon Bay down to the very south bay. We also sampled outside the Golden Gate in the open ocean within the 3 national marine sanctuaries that are sitting outside the Golden Gate from us. And what we were really out there looking at was we wanted to understand the differences between wet and dry sampling. So what we did is we monitored each site once during the dry season, and then we came back to each site just after storms. And we did this sampling on the Derrick M. Bayless and another boat at the from the Baykeeper. So what we did was, let me move on because I'm getting nervous. So I'm sorry. I'm really excited to be here, and I'm so excited that I'm a little bit nervous right this second. Thank you. A lot of effort has gone into this project. All right, so backing up, I'm going to repeat what I said on the Derrick M. Bayless. So our research was done using 2 different vessels. The vessel, the Derrick M. Bayless, is a beautiful sailboat, and we were able to get out into the sanctuary and throughout most of the bay on this boat. But in order to get to the very south sections of the bay, where it was really shallow, we ended up having to use the Baykeeper's boat. And again, we monitored each of those sites at different times to understand the differences. And what we used at each site was a number of techniques. So we used different techniques to look at different size fractions. But for this talk, I'm going to focus on the manta trawl sampling. This has a long history of use, especially for microplastic sampling, and this is a well-used technique to analyze 1/3 of a millimeter and larger. For all of the samples throughout the project, we analyzed fragments, foam, film, and spheres. And then for a portion of the samples, we analyzed fibers. And the reason that we subsampled for fibers was just the sheer number of fibers, but also there's a growing consensus or there's a growing questioning from the science community at how accurate are these, is the mantitrol to analyze fibers. So the way fibers are shaped are long and skinny, and so they often can fit through the mantitrol net, possibly underestimating the fiber count. So now, looking at the results, which is really exciting. So this is a figure of each of the samples that we took throughout the project. This is the dry weather results. So we wanted to understand the difference between the bay and the sanctuaries. And what we saw, even with these dry results, is that there is a difference. And once you add in the wet weather results, we could see that there was a 30 times more plastics in the bay samples than we were seeing in the sanctuaries. So this is a pretty big difference. We did somewhat expect this, but we now have the data to support this. And then looking at the wet weather sampling, we saw a really high variability throughout the sample sites, both in dry and wet weather. But on average, we could see 10 times more microplastics in the wet weather sampling, samples over the dry weather samples. So this is comparing only the bay results. When we're looking in the sanctuary, we don't see a significant difference between these two data sets, and that's probably because the counts in the sanctuary, they're pretty low to begin with. And so now looking at how these results compare to other areas, so we're looking at other researchers' results, we're still seeing that San Francisco Bay has high levels of microplastics. So you can see that we have 2 bars for San Francisco Bay. We have our current study in 2019, and then we have the study that Becky and her team led up in 2016. Well, the field work was in 2015. But you can still see that there is a difference between those 2 different data sets. Our data set has many more samples, and we did sample in the dry weather season, so that's it's giving a better representation of the entire sort of bandwidth of the samples. But overall, you can see that San Francisco Bay has higher levels. So comparing to the Baltic Sea, we're about 7 times higher potentially. And you can look and see the differences between the other areas that I mentioned. So looking at what types of particles are in the samples, we did see a lot of fibers. So 74% of the particles were called out as or identified as fibers. A pretty good number of fragments were also found in the surface water samples, so 18% of them, followed by foam, film, and spheres. And then, as Meg mentioned, one of the really important parts of this project is trying to understand and identify how much of these particles are plastic. So looking at all of the particles, we can generally say that more than half of the particles are plastic, but now I'm going to look at the specific particle types. So looking at fibers, the University of Toronto helped us identify that 53% of the fibers are plastic. And you can see that there's a percentage, close to 20% are also identified as anthropogenic unknown. And as Meg said, this could also have plastic particles in it. Most of the polymer types that we're seeing in the fibers are polyester and acrylic. So looking at fragments, these particles, 87% of them were identified as plastic, with most of them being polyethylene and polypropylene. And then polystyrene foam bits. We saw these particles in our surface water samples, and we saw very few in the other sections. But what we're seeing, 68% of these were found to be plastic, and most of them polystyrene. But we also saw that there were less than we expected in many ways in the surface water samples. We know in San Francisco, many of you are out on the beaches doing work there, that we know that polystyrene foam, or we know that foam is on our beaches. And it was a little bit misleading to not see as much in the surface water samples. But I think if you think about foam being so lightweight and mobile, that there's more to this story to be told. So, but we did see it in our samples, and I think this is good results to support policy action. So this is moving into sort of policy action side of things. Like Meg said, we can use the particle type and the plastic percentage to really understand what the source is upstream. So we can look at these fragments and try to tie them to the breakdown of single-use plastic items. We can look at these fibers, polyester fibers, and try to link them upstream to clothing, bedding, carpets, and so on. So with that, I'm going to turn it over to my colleague Diana Lynn, and she's going to speak about sediment and fish.
So looking at microplastics in sediment and fish is really important because they're the foundation of the food web in the San Francisco Bay. So in this results for sediment and then fish. Most of us would expect to see microplastic on the water surface, just as Carolyn just described. But some plastics also sink, and even many of the plastics that initially float will eventually sink as biofilm and other particles start collecting on these particle pieces. And over time, microplastics can accumulate in the sediment. This is the first study to look at microplastics in bay sediment. And so before this, we had no reference for what potential levels could be. In our study, we collected samples throughout San Francisco Bay as well as in Tamales Bay. Tamales Bay is next to the Point Reyes National Seashore and far away from urban populations. And so samples collected in Tamales Bay were used as a reference or comparison for samples that we collect in San Francisco Bay that is surrounded by urban populations. The concentrations we measured ranged from 0.1 microparticles per gram to 60 microparticles per gram. And you can think of a gram of sediment as being less than a teaspoon scoop of mud. We saw lower concentrations in Tamales Bay compared to San Francisco Bay, and the highest concentrations in Lower South Bay. And Alicia later will describe more about wastewater, stormwater pathways, and the Lower South Bay is heavily impacted by these pathways. We saw the let's see, what was I going to say? And you can see this by the size of the circles. So the concentrations in the Lower South Bay were high, not only compared to San Francisco Bay itself, but also high compared to studies around the world where microplastic and sediment have been studied. So just to name a few examples of the other studies, Tampa Bay, Florida, Baltic Coast in Germany, as well as Jiuzhou Bay and South Yellow Sea in China. So what type of microparticles did we see in the sediment? This is a pie chart showing the breakdown of the different shapes that we identified in all of the sediment samples combined. 15% were further analyzed by spectroscopy to confirm whether they were plastic. Of the fibers, which represented a majority of the microparticles, many were identified as polyester and acrylic fibers, and textiles could be is a potential source of these. Among the fragments, many were black and rubbery, which we suspected was rubber based on the compressibility, the color, and the texture, but we couldn't 100% confirm this from the spectroscopy results. These type of particles were also abundant in stormwater samples, which Alicia will talk more about later. And so a possible source of these rubbery fragments is tire wear. A few of these particles were sent to another lab for analysis using a different instrument, using pyrolysis GCMS, and their results came out that indeed the particles that they analyzed was tire wear. And Chelsea Rockman will talk more about this really exciting result later. I don't know about exciting, depressing, I don't know. Interesting. The remaining particles were film, foam, and sphere. Interestingly, the sphere particles were mostly clear polystyrene spheres, which have many different applications, including particularly in the water purification. For example, they're used in water softening equipment, as well as in the biotech industry. So one example might be biomolecules can be attached to these spheres in clinical and research studies. Now you can see it because it was clear. So once these plastics are in the sediment, they can be a source to the food web, starting from small worms and other organisms that eat the sediment, which are in turn eaten by small fish, which are in turn eaten by bigger fish. And so in this study, we sampled for prey fish throughout San Francisco Bay, and we collected both tossed smelt and anchovies from both sites. These species are really important to the bay ecosystem because they are eaten by everything that's bigger, essentially, which is why they're called prey fish. We collected samples in Tamales Bay for comparison to San Francisco Bay. And, sorry, let me backtrack a little bit. To San Francisco Bay. What we found was that many of the fish had ingested microplastics and microparticles. And fish from the San Francisco Bay had more microparticles than fish from Tamales Bay. We also found that tossed smelt had more fragments than anchovies. And a possible reason for this is that their preference for where they like to live. Tossed smelt prefer to be in shallow margin areas of the bay versus anchovies tend to prefer more open areas of the bay. This pie chart shows, again, the breakdown of the different shapes of microparticles that we saw in all of the fish samples combined. And they were mostly fibers. 20% we were able to confirm were plastic, and many were in the anthropogenic unknown category, which Meg mentioned means that they were dyed, but we couldn't confirm whether they were plastic or not. The levels that we saw in the fish from San Francisco Bay was similar to other studies around the world of other small fish. This includes in the Mediterranean Sea, the Adriatic Sea, as well as in Shanghai, China. From these results, we know that microparticles and microplastics are entering the bay ecosystem. And as Suzanne Brander described earlier, we know that there are potential impacts to organisms that either ingest or are exposed to microplastics. But these thresholds are still being studied. And so we still don't know what are potential impacts to the bay from the results that we've presented so far, the levels that we measured in the fish, the sediment, as well as in the surface water. So next, my colleague Alicia Gilbreth will talk more about how these microparticles and microplastics might be getting into the bay through wastewater and stormwater pathway.
Hi, I'm also really nervous. Thank you. Thank you so much. Okay, so how are microplastics getting into the bay? Well, the two major pathways are wastewater and stormwater. So wastewater is everything that goes down your drain, your toilets, your laundry, and it goes to a wastewater treatment plant where it's treated, thankfully, and then it's released out into the bay. So we're really interested in the microplastic concentrations in that treated wastewater. Stormwater occurs when rain falls on the landscape, it runs off into creeks and storm drains, and that stormwater flows out into the bay. Mostly in our region, it flows out untreated. So we're really interested in the microplastic concentrations in that untreated urban stormwater. So what we did for wastewater is that we looked at 8 facilities around the bay, and 4 of these wastewater treatment plants were secondary treatment plants, which is a more traditional level of treatment, and 4 of them were a tertiary level or advanced filtration. Together, these 8 facilities discharge about 70% of the total wastewater, treated wastewater to the bay. For stormwater sampling, we looked at the 12 locations that are indicated by the black dots. Can you see those? Okay, well, they're attached to the colorful polygons, and those colorful polygons represent the watersheds. That's all of the landscape that drains to our sampling points. So for example, this is the Coyote Creek watershed, that teal polygon, and all of the landscape in that polygon drains to our sampling location on Coyote Creek. We sampled during a single storm event at each of our 12 locations, and we sampled over the course of the entire storm event. As you can see, they vary geographically, in size, and also in the land use character that makes up those watersheds. So what do we see? Well, unsurprising given what we see in surface water, sediments, and fish, we found that fibers and fragments really dominated in wastewater and stormwater. In wastewater, we saw many more fibers than fragments, and in stormwater, we saw more fragments than fibers. I'm going to give you a high-level finding from each of the pathways. We'll start with wastewater. So here you're looking at the concentrations from each of the 8 facilities, and when we group them between secondary treatment plants and advanced filtration, you can see that the advanced filtration plants release much lower levels of microparticles than the secondary treatment plants. Shifting over to stormwater, we found that half of the particles were those rubbery-like fragments that Diana just spoke to and that Chelsea Rockman is going to speak to later today. So how many were we finding? Well, in stormwater, these are the results from the 12 watersheds that we studied, and they ranged in concentrations between 1 and 30 microparticles per liter. Generally, the more urban stormwaters had more microparticles per liter, and the less urban ones had fewer microparticles per liter. The one land use that really tracked well with the microparticle concentrations was industrial areas. So we're talking about factories, manufacturing, old military areas, et cetera. As you can see on the left side of the graphs, the watershed with the highest industrial area also has the highest microparticle concentrations, and on the right side of the graph, the converse is true. It's obviously not a perfect correlation, but it's both strong and significant. When we compare the average concentration of microparticles in stormwater and wastewater, you can see that it's much greater in stormwater. And in fact, when we extrapolate our data to all of the wastewater going into the bay from the entire region, we estimate that there's 17 billion microplastic pieces to the bay annually, 17 billion. When we extrapolate our stormwater data, we estimate 7 trillion. So we're talking 300 times we're talking stormwater's 300 times the load of wastewater. So that's pretty depressing. But I'm not going to leave you there. Mark spoke earlier to what he called LID, or we call LID, or Green Stormwater Infrastructure. What you're looking at here is a rain garden in El Cerrito, California, that we got to study. And what's happening here in this picture is it's raining, water's flowing off the landscape and into our rain garden. A cross-sectional view of that, water's flowing in at the curb. It ponds on top of our rain garden and filters down to where it finally flows out an underdrain. We got to measure microparticle concentrations at the inlet and the outlet to this rain garden. What we found is that the rain garden reduced microparticle concentrations by over 90%. So this is one of the really elegant solutions to reducing microparticle and microplastic concentrations to the bay. I am nervous. To the bay. My colleague Carolyn is going to talk to more solutions later today, so stay tuned for some more optimism. With that, turn it over.
So I hope this was a good summary of our very comprehensive study of microplastics in the bay. I think some of the takeaway points are this pollution is pervasive, very widespread. We see it everywhere. And this very interesting finding that Alicia presented, where stormwater actually has much higher levels than wastewater. That these stormwater findings are very significant because folks hadn't really been looking at urban stormwater before. We've studied wastewater, but the stormwater findings are quite new and intriguing. So I'm wondering about questions from the audience. If anyone's got any follow-up questions, big picture or more technical, for my colleagues here today. Jen?
Hi, Jen Jackson, City of San Francisco. Could you talk a little bit more about the polystyrene spheres?
Sorry, would you mind?
Hi, Jen Jackson, City of San Francisco. Could you talk a little bit more about the polystyrene spheres? I think you said that they were associated with water softening equipment and pharmaceutical industry.
Yeah, most of the spheres that we saw were clear polystyrene spheres, and potential sources based on our literature review are from water purification. And I gave one example of what type of equipment it would be used in, as well as in the biotech industry. I don't know if I can say more than that. Atlas, do you have anything to add? No? Okay.
I would also note, we did see some polystyrene spheres in the surface water, but more of the spheres were polyethylene and polypropylene. So the polystyrene spheres tended to concentrate more in the sediment. Yep. Please raise your hand. Yeah, our mic runner's coming.
Hi, I'm Asa Bradman at UC Berkeley Public Health. I have two related questions. One, if we look at the wastewater and stormwater, do you have a sense of the kind of the dominant contributors? I assume fibers maybe in the wastewater is coming from laundry sources or others. Do we have a sense of air contributions? You know, what's being deposited that's being carried out? And do you have any idea of those proportions? And then this might be a question for this afternoon, but if we're using, for example, the rain catcher as kind of a filter, what's happening to the soils and where is the material going? Thank you.
That's a really great question. I think as far as the fibers goes, it's really hard to link it back to a particular source. I think you bring up a really good comment on one of the things that we saw. I mentioned that we developed rigorous field methodologies. One of the things that we did and that was somewhat unique in this field is we did field blanks and we did field duplicates. So for those of you who work on legacy contaminants, this is nothing new, but it is for the world of microplastics. And we saw fibers in our field blanks, which to us spoke to the ubiquitous nature of, you know, sort of the general fibers around our daily lives, what I refer to as the pigpen effect of just as you move, we generate clouds, whether it's in an industry or an individual. So we believe that there are other pathways out there than the two that, can you tell I was nervous too, but I'm also excited. We believe that there are other pathways out there, and I think air deposition is a really important one.
Okay, and I'll speak to your, can you hear me? I'll speak to your second question about what's happening to those microplastics that are going into the soils of our green infrastructure, rain gardens. This is a really important issue, not just for microplastics, but for other pollutants that we're interested in capturing, and even the pollutants we're not as interested in capturing. To my knowledge, we are not yet working on those solutions, or we're at the beginning stages of working on what those solutions are going to be. I think it's really, people in the field are really well aware that this is an issue, and we're just right now trying to get green infrastructure in the ground. You know, there are potential solutions such as scraping the top layers, which is where most of the pollutants and probably microplastics are depositing. So we can scrape those top layers and replace them. Right now, we're not working on that issue quite yet. Yes.
I just wanted to follow up on that and say that this is something to think about, is because the rain gardens are obviously working to remove the particles before they enter the bay, but really they're still there, and we're going to dispose of them somewhere else. So really we want to look up, you know, further upstream to address these solutions.
Yes.
Hi, Olivia Angus from Surfrider San Francisco. I have a question for Caroline. You had in one of the slides with the pie charts, it said like plastic particle, anthropogenic, non-plastic, and then a non-plastic pie slice. Sorry, now I'm getting nervous. But the title was talking about polystyrene foam. So what's an example of polystyrene foam that's not plastic?
Oh. That's a good question.
I think the pie chart was referring to all the foam particles, and the polystyrene foam was the type of plastic that we spotted in the plastic component.
So it would be I think it was, I can't remember the percentage, 68% was plastic of the polystyrene, of the foam particles, and then the other particles that are other foam particles that are non-plastic. Maybe Alicia can help us. Yeah.
I can't really speak so well to surface water, but I can tell you in wastewater, we found foam in the wastewater effluent, and that tended to be soap scum. So it was a kind of a foam detritus. So that would be an example of a foam particle that would be, based on the visual observations, classified as foam. But when we actually do the analytical work, we can say it's not a plastic polymer.
Yeah, and there are other natural occurring, let's say like plant debris or other things that may look like foam when you're picking it at such a small level, but then those are showing up as natural once you run the tests. So I think the big, we can look at that in more detail to try to figure that out, but we classified them just as non-plastic and plastic. So there's more questions that we can, look through the data and understand.
Oh, and there's other foams that are plastic too, like polyurethane. But polystyrene is the big one.
Sarah Huber with the State Water Board. Since this was the first time you were addressing stormwater and microplastics in the sampling mechanism, can you just describe a little bit more about your stormwater composite sampling methodology or something related to that?
Yeah, sure. So we. Okay, sorry, I can't tell. Closer. Okay, cool. Thanks for that question. So we drew on methods that we've been using for the regional monitoring program and SFEI for many years now, and they're pretty standard methods for sampling pollutants. But we did have to develop or modify our methods that we typically use, and they differ from what's typically being used in the literature so far for stormwater or for water sampling, like river and creek sampling, for other studies of microplastics. And the reason is that most other studies are sampling the very surface of the water, and they're using larger sieve sizes or mesh sizes. So we not only used a smaller sieve size to try to capture more microparticles, but what we did, getting to your question, is that we moved the tube from which we were drawing water from up and down throughout the water column to try to characterize the entire water column. And what we did is we composited over the course of an entire storm. So when I say composite, what I mean is that we take a sample at the beginning, and then an hour later we take another sample, and an hour later we take another sample. And so we do this over the course of an entire storm event at each of our locations. Does that address your question? Or do you have more?
So I think one of the questions I had was also, what kind of tubing did you use, or did you have any potential modifications to materials in order to prevent cross-contamination possibly, since plastic tubing seems to be a very common stormwater sampling protocol?
Right, and how problematic to be sampling for microplastics and using plastic tubing. We did use pretty resilient kind of tubing, Teflon tubing, and the tubing in the pump head is a silicone tubing, and it was studied by some, I believe, Chelsea Rockman's colleagues or other, Paul Helm, excuse me, up at Ministry of the Environment, Canada. Yes. Thank you. And so they did some work to study what sort of contamination there might be from using that particular tubing, and they found no contamination. So we feel pretty confident that there was none or very little contamination caused by the methods that we were using. But real quick, to address the size of tubing that we used was a 3/8 inch diameter. So I can see that you were looking for that information. And that may have limited how large of microplastics, of plastics that we were sucking in through our tubing, but it should have still been capturing the majority of microplastics. You bet.
I'm Jess Daniels with Fibershed, and so I heard that fibers are the most common microplastics you found in the Bay Area, and the majority of those are plastic fibers, which correlates, or I suppose mirrors, global fiber consumption, which is 60% synthetics. So I think that's interesting. But I was curious, Carolyn, if you could talk about the manta trawling, and if you have any way of assessing if that is underestimating fiber count and pollution levels.
There are some, we can refer you to additional research that is looking into that. But for our project, I can speak a little bit about the blank contamination that potentially could elude that, there's a lot of fibers out there and we need to do potentially a better job at analyzing them. So we did in the project look to a couple other techniques, and at each site, we attempted to do a pump system similar to what happened at the stormwater, which is actually a really exciting finding coming out of, or process coming out of the project is this sort of exploratory methods. And so that ended up not being a great method, but we also took 1 liter samples that Chelsea's lab has analyzed, and some of the data is in, or some of the results are in my chapter, or our chapter. And I guess the question for you for the fibers is, we see quite a bit of fibers sticking onto the net, for one thing, and we know just by the shape and size that they can fit through, so it is potentially underestimating it. And I think we're gonna have to follow up with you on some of the actual specifics of the other projects looking at estimating, understanding how good they estimate fibers.
Hi, I'm Nicole. I was curious on that note about fibers. It seemed like across all the samples, fibers were the largest portion of what you guys collected, and fish seemed to be the only sample taken where the anthropogenic unknown was the highest proportion. Have you guys had any hypotheses around why that is, or maybe how it could relate to consumption in humans higher up?
I'm actually going to, Alice, did you want to speak to that, or?
Well, I think we have a lot of anthropogenic unknown throughout all the samples, but we have like so many in fish because most of what we found in fish was fibers, so it seemed like a big portion of it was unknown, but really they were everywhere. And the reason why they exist is because of dyes in the fibers. So our Raman instrument, it detects the dye, but it doesn't detect the polymer. So the dye masks the polymer, which is why it's difficult to identify. But really, this problem was apparent throughout all the samples.
I think I can speak loud, but I would also just add that when you're taking it out of a fish, it's a gunky sample. And so the fish were the dirtiest of the samples, and they were the small particles. So we had to use Raman for all of it. So I think that's also one of the reasons, and I would say that you probably saw that for the manta, when Carolyn was talking about surface, for the microfibers, we had less of the unknown and more of the known, and that's because the fibers were big enough that, Alice, I think you used FTIR for all of them, right? So Raman does worse at the microfibers than the FTIR, but when the particles were so small, we in our lab were limited by Raman. So the smaller the particle, the gunkier they are, which is the fish, the more unknown we have.
So that was Chelsea Rockman, who led the analytical work with Alice, and so I would definitely direct technical questions at the break to those two. They're going to be able to answer all your questions.
Hi, I'm Emily Rush. I'm the executive director of CalPERG, the public interest group, and I'm still wrapping my head around this idea around the rubbery particles being from tires being such a large percentage of what you found and what that means. And I apologize if you touched on this already, but based on the few other studies that have been done in other parts of the world, does it correlate, or is it just they didn't discover them, or is this something we truly think is unique to the San Francisco Bay because of geography, et cetera?
I have a hypothesis that I was thinking about last night. Would anyone prefer to take it on? Okay, brilliant question. What I what I think is going on, and I spoke to this earlier about our methods, is that we were sampling throughout the water column, whereas most studies, they're sampling on the very surface. So they're biasing towards the more buoyant particles. Well, these black rubbery fragments they're denser, and so they are within the water column. I mean, the water column is very turbulent, right? So they're mixing up, but they're not exactly floating. So I think that's why our study we found so many, and why they're not seeing them in other studies around the world. And I believe because tires are everywhere, right? Every urban area, and they are studying urban areas elsewhere. But I think what we'll find is as they adjust methods, which I think they will, I think our study will set a precedent for monitoring methods. We will be seeing more black rubbery fragments, I believe.
Yeah, I totally agree with your hypothesis. I think studies should be like the San Francisco Bay study. It should be holistic and sample multiple various matrices throughout the ecosystem. And also, this is one of the few studies that study stormwater. It's actually a very novel pathway that not very many studies sample. So car dust is everywhere. It's just we haven't been studying them.
Yeah, that's exactly right. There's just not that many studies yet for us to compare to, but there are studies that have been published that do see these rubber tires. And as I just described, and Chelsea might talk more about later, these rubbery fragments are really hard to identify as rubber based on their small size, their texture, and compressibility and everything. But there are other studies that have detected them.
I was just going to point out that a lot of the stormwater studies have been looking at trash, which is defined at 5 millimeters and above, so it's really missing that smaller size fraction. So that's why we have limited studies on microplastics in stormwater. And there isn't a sort of given method, so hopefully we can start developing methods that can speak to each other. So.
Yeah, Alexander Black, microfiber solution question. I'm looking at a study that just shows there were just under 500 releases of raw sewage between last fall and this spring in the San Francisco Bay Area. Just wondering if you can speak to any types of concentrations of fiber in sediment, particularly geographically when you think of all these wastewater treatment plants and sewage runoff, and just microfiber release in both their sewage sludge and also in their effluent. And if there were ever a study, as you talk about later, I guess today, there's going to be a discussion of a filtration study perhaps to be applied to the area, just looking at source points, that type of thing.
Well, our study was focused on treated wastewater, so we didn't examine untreated wastewater. You do bring up an interesting additional pathway, not part of the study, which is that the treatment in the wastewater facility doesn't delete these plastics, right? It pulls them out of the water column, but it often means that they're building up in your biosolids, a waste product, which can be transferred to agricultural landscapes for nutrients, right? So we may actually be grabbing plastics from wastewater as we treat it and moving it to a different part of the landscape. That might not be the solution that we really want. So this actually speaks to some of the solutions discussions we'll have this afternoon about where we should try to capture microplastics, earlier in the sort of flow of plastic, the better.
I was just going to build off of that with, so the untreated sewage would be overflowing in San Francisco. So if there's too much rain, it'll just overflow without being treated. So in that material, that would be going straight into the bay or into the ocean. But we do in the surface water chapter, which is getting at that sort of part of the question, that would look at, you could see in those results how many microfibers per kilometer square. So this is the abundance. So the results are in there showing I have on average in the bay during wet seasons, it's 580,000 microfibers per kilometer squared, which is a really high number, but you can see how we compare to other areas. And there must be better ways to sort of describe that number so that it's more powerful. So.
All right, I think we're going to have to wrap up now. Well, luckily, it's break time, so we can all have some snacks and some coffee, and come grab us if you have any more questions.